My study is part of the TACTI-002 trial. This is a phase 2 study in which eftilagimod, that is a soluble LAG3 protein, is combined with pembrolizumab in different cohorts. And in this congress, we present the part B of the trial, in which patients receive this combination when they have progressed to the combination of immunotherapy plus chemotherapy or immunotherapy alone. The study design is a phase 2 trial in which there are 3 cohorts. The first one is patients who are treatment naive. The second one is part B, which is the one that we present in the Congress, that includes patients who have progressed to immunotherapy alone or in combination with chemotherapy. And the third one is patients with head and neck carcinoma that have progressed to standard chemotherapy. In this part B study, patients need to have a confirmed progression with 2 consecutive CT scans, and they receive treatment with eftilagimod every 2 weeks during 8 cycles, and then every 3 weeks until 1 year, combined with pembrolizumab. And after this year, they receive pembrolizumab alone to complete 2 years of treatment maximum. The primary endpoint of all these cohorts is objective response rate per iRECIST criteria. The key results are related to the efficacy. This study includes 36 patients, all of them previously treated with immunotherapy plus or without chemotherapy. The majority of patients were PD-L1 negative or PD-L1 low expression. And what is important to underline is that in this population, we found an objective response rate about 8%, with a disease control rate of 33%. We have to take into account that these are patients that have already progressed to immunotherapy, with a median overall survival of almost 10 months and a progression-free survival of 2 months. But what is important also to underline is that the 6 months PFS rate was 25%, and the overall survival rates at 12 and 21 months were 44 and 39% respectively. As our population was predominantly patients that have already progressed to immunotherapy, and we observed that those patients with high PD-L1 expression and those that have already responded to the previous treatment were the one that most benefit to this strategy. I think that these treatments open a new door for new potential strategies for patients that have already progressed to immunotherapy. These are a preliminary data, is in a small cohort of patients, but this data supports further clinical investigation to innovate this treatment in patients that have already progressed to chemotherapy.